Now in detail, Lycopodium distribution occurrence. Before that, Lycopodium, commonly called as club moss, ground pine, trailing over green. These are the synonym of Lycopodium. So far, represented 400 species worldwide. In India, they are represented by 33 species. Indian species are Cernum, Lycopodium clavatum, Lycopodium serratum, volubile, inundatum, Figmeria, Selago. These are the common Indian species. Now let's see habit. What is habit? Natural place of an organism. Now tell me the pteridophyte habit. Pteridophyte
Marshland.
are true land plants. That's why these are the found open woodlands and moist acidic soil. As they are true land plants, they are openly found in woodland, moisture places, acidic soil, and some of the lycopodium species grow as epiphytic. Example, Pligmeria. What is epiphytic? Epi means upon. Phytic means plant. Instead of attachment to the soil or land, they can depend on other plants. On other plants, these are called epiphytic, and they are appear as a erect and shrub-like structure, creeping, erect branches. So they are the different forms of lycopodium. Now, if you see the lycopodium structure, yesterday we discussed about this lycopodium specimen. Hello? Yesterday clearly discussed about this lycopodium specimen. Lycopodium may be horizontal like rhizome in this diagram like this, or erect stem, or both are present like this. So this is the sporophytic body, and this sporophytic body distinguished into three parts: root system, shoot, and leaf, green color leaf, like this. These are all lycopodium species. Let's discuss one by one. After root actually branched adventitious roots. Stem dichotomously branched. Stem dichotomously branched. And external morphology. The plant body belongs to, hello? Main plant body.
Yes, sir. Plant body is a sporophyte.
Right. Plant body, sporophyte. And this sporophyte differentiated into?
Three parts.
Right. What are those?
Uh, Root, stem, leaves.
Right. And these leaves and actually considering the variation of habit of different species, these are divided into two subgenera. The two subgenera of Lycopodium, Eurystachya, Truffulostachya. Let's see one by one. So based on the habitat or structure, these are two types, Eurystachya and Truffulostachya. First, Eurystachya Lycopodium species erect or pendant. Maybe they are erect in nature like this. Let me take pen. Maybe these are maybe like this erect nature, or maybe from the substrate they are pendant like this, maybe pendant. So two types. But in Raphalostrachia, these are prostate, but not erect and not pendant. These are prostate like this, like a rhizome. It consists rhizome and it shows continuation growth horizontally. So for prostate and stem with upright branches, but the main trunk is like this prostate, but the branches are upright. And we can see diagram also like this. These are the branches, and the branches also dichotomous branches. Here also same. Mainly the dichotomous branches clearly appear in Aristachia. But here dichotomy noticed first formed branches, but later monopodial. Actually starting dichotomous, but later it continuation as a monopodial like this. Only one side, monopodial growth. Leaves and sporophyll most of same size. Leaves, normal leaves and sporophyll leaves, same size. And here actually forgot one thing. Roots arise only from the base of the stem, only from the base of the stem, because it is erect one or pendant one. That's why, for example, if roots are present from base, like this, from base roots are arranged. But this, here the same roots, already told you roots are advantageous, but here advantageous roots, the not base, entire length of the stem arises from entire length of the stem. As it a prostate one, entire branch, that mean here branches, root system, root. So these are the roots, center system. Here another one, leaves and sporophylls almost same size, but here sporophyll smaller than foliage leaves. That is the difference in Raphalostochia and Eurostochia. And a vegetative reproduction takes place by bulbils. Uh, the sporophyll arrangement actually is not given any vegetative reproduction, mainly vegetative reproduction present in Eurostochia. Sporophyll arranged form the cone strobili. That means the main reproduction takes place by cone or strobila in Raphalostochia. So these are the based on structure of the lycopodium. They can classified into two parts, two subdivisions, Eurostochia and Raphalostochia. Right. So this is important for short answers in theory. In practical, we don't write all this. In practical, just only one sporophytic structure. It consists root, shoot, stem, uh, root, shoot, and leaf. This much enough. Next,
Sir, for theory, what is important? You said.
a theory. There is no particular. That means what I'm saying. Already told you during practical classes. The first of all, you should learn the concept of
Yes, sir.
bryophyta, concept of pteridophyta, gymnosperms.
Yes, sir. General characters.
General characters, right? The general character can applicable to each and every type studies. After learn the general character, you can that mean modify the some type studies. For example, if you take bryophyta, bryophyta general character,
Yes,
and
sir.
while you read about the type studies like Markansia, Anthocyrus, and Funaria. Just write down side by side, one side Markansia, Anthocyrus, and Funaria, and just compare each other.
Okay.
Compare each other. It would be easy to you, right? Don't focus on what are the important questions. So it is best, but try to develop the subject. Okay.
Sir, thank you, sir.
So if you don't have enough time, you can select any two black. For example, your aim is only pass marks. You don't want any subject. You don't want any concept. In that situation, if that's so, you just select any easy two block, any two. Actually, in our theory book, four blocks are there, right?
Yes, sir.
Out
Tally
of
bot.
four, right? Out of four, you can choose any two blocks. That was enough to get almost 50% marks. 50, 50 marks from this. If you select any one block, you can pass easily. That is your choice. But my suggestion is don't focus on important question. Just focus on subject, right?
Okay, sir.
Uh, it is where it is. Right. So these Erostochia, Aristochia, Truffalostochia, this is only for theory, but not practical. Okay. Right. Now next one. Now Lycopodium. Let's discuss one by one. Root system clear. Uh, uh, Adventitious roots. You can see here adventitious roots from the pericycle. That is not an important one where they are arranged, but only adventitious roots enough. Theory and as well as practical also. A stem, how would the stem in Lycopodium weak, slender, rhizomatous, erector, pendant, creeping, and others. Branch basically dichotomous, sometimes monopodial. Stem branches covered with leaves. Already they are all discussed. What about leaf? Leaves are simple, sessile, scale-like, small, with median, vein, microsporophyll, spiral arranged, and dense of stem, decussate, or oral arrangement. Decussate, but it's very rare, but mainly spiral arrangement. Decussate arrangement means simple, the opposite direction. The leaves arrangement, opposite direction, that is called decussate. And here are the two types of leaf in Lycopodium, isophils and anisophils. The name itself given isophils and anisophils. What is iso? Hello?
Same.
Same, similar. All leaves are of the same type. That is iso. Aniso means different type of leaves. For example, here the given Lycopodium volubile, four rows of leaves. Two lateral leaves are large and falcate leaves. One ventral row small air-like leaves. Dorsal row medium size. So if the that is based on the dependent on the species, the maybe the leaves are isophils or anisophils. Iso means all leaves are similar. Aniso means different shape. So you can see here the stem with leaves and roots, dichotomous, branched, adventitious roots, stem with leaf in diagram. Here the anatomy of root, it is neither important in theory or practical. So don't focus on this important anatomy of root. But anyway, the fundamental remember, if any anatomy, there are three regions, epidermis, cortex, and stele. That much enough. Okay. And this is important. Anatomy of stem. It is important for theory and practical also. Already you have section cutting, right? Hello?
Yes, sir.
Yes. The section cutting material, if the examiner give the lycopodium stem, you have to make a section and observe the microscope. If you take a section and observe the microscope, you can see some characters. Let's discuss one by one. I already told you if any, excuse me, any uh, internal structure, there are three regions. What are the three regions? Hello?
Epidermis,
Right.
cortex, stele.
Right. The three regions. Well, let's discuss one by one. Epidermis, that is common character. Epidermis never change. Always epidermis uniseriate, protective layer with thick outer cuticle. This is common.
No
Outer layer.
interspaces.
Yes. No interspaces also. No inter. That means no intercellular space. Without gap, the cells are arranged. And you're not given, but remember, uniseriate. Almost epidermis single uniseriate, made up of single seriate and covered by cuticle. That is common. If you take root also, stem also, anyone. Cortex. Nature varies with species and stem diameter. Homogeneous and heterogeneous. Simple. Homogeneous means entirely parenchymatous. In if you take the lycopodium uh, section cutting, in some species the cortex entirely made up of parenchymatous, or the clearly three zones are present: outer chlorenchymatous, middle parenchymatous, and inner sclerenchymatous. If it is made up of different composition of tissue, that is called heterogeneous. Same same means either parenchymatous or chlorenchymatous or sclerenchymatous. Anyone, any similar group of cells, that is called homogeneous. Next, after this, the end of the cortex is endodermis. This is the distinct in young stems. This is also actually not given. Endodermis similar to epidermis. Similar means endodermis also like epidermis, single layer without intercellular space. And here the stele type is protostele. Proto means hello. Proto means
Sir, proto means the xylem and phloem are around each other.
that is the steel composition. But what exactly proto means? Steel means it is the center position and which
Yes,
is made up of xylem and phloem. But what is proto? Proto steel.
primitive.
Proto means actually the first. General
Yeah,
definition.
primitive.
Proto means first or primitive. Okay, that means these are the first formed steel. Let's see how the first form. The already told you first vascular system developed in pteridophyta, right? That's why these
Yes,
are
sir.
protosteel. Let's discuss about the protosteel here. Inside the steel have xylem and phloem. Protosteel means primitive steel. There is no pith. For example, if you take higher plants steel, higher plant steel clearly higher plant steel, that well developed steel. This is steel. And let's change color. Vascular bundle as a round. Vascular bundle as a round. And central this region middle. Sorry. The center region middle are with this one. This is advanced steel. But in proto steel, what happened? This middle are absent. This middle are with absent, no middle, no middle, and no ring of vascular bundle. Here the clear ring of vascular bundle. Inside the vascular bundle, Each vascular bundle, for example, if you take a vascular bundle, a cambium present between xylem and phloem, outer side phloem, inner side xylem. So this is arrangement. But what happened in protostele? Only xylem and phloem present. There is no cambium, there is no pith. For example, haplostele here. This is haplostele. Haplostele means simple, without middle or pith. First haplostele, center xylem, simple, surrounded by phloem. So this is haplostele type. No pith, this is important. No pith at the center. Xylem forms central core surrounded by phloem. This is surrounded by phloem. Central core, this is center position and surrounded by phloem. Right now, this type is called haplostele. Xylem is exarch condition. Types of protostele may be the based on species, actinostele, plate-like plectostele, or mixed protostele. Let's
Sir,
discuss
what
one.
is exarch?
Exarch means, for example, in xylem, this is steel. Xylem formation in inner side. Inner side means this is center. One xylem cell, second xylem cell, third. So in this first formed xylem cell, toward the center. If the first formed xylem, like this, for example, let me draw clearly. This is steel, for example. This is center. Xylem, small xylem, if present center, like this. That is ender condition. If the small reverse, that means first form xylem outside and development toward the center. After formation xylem, anyway, after formation xylem, development outside to center, that is called exarch condition. Hello?
Okay, sir.
Right. So simple development of the xylem. Outside to the center, exarch condition. Center to outside, ender condition. That's it.
Okay, sir. Thank you.
And another type is actinosteel. Actinosteel xylem star shaped. The name itself given actino means star shaped. Like this. This is star in this diagram. Like this. This all xylem. And surrounded by the star shaped is xylem. And rest of the place is, hello?
Phloem.
Right, that's it. And next one, plectome means plate-like. So most probably in our section cutting, have you done section cutting today? Sir have explained or not?
Yes, sir.
Right. If you observe after section cutting, if you observe, actually, what are given in our university, the material The section cutting material along lycopodium is plectosteel like this. This type of arrangement. If you make a section, if you see under microscope, the steels appear like this. Plecto means plate like. That means xylem. Next xylem. Next xylem. In between phloem. Between phloem cells are present. So this is plectosteel. And mixed means actually we don't know where the xylem, where the phloem. They are irregularly mixed like this. This is the mixed protosteel, right? In examination point of view, you can write mostly, but in theory, you write actinosteel and plectosteel mixed steel. But in practical, one enough. That is plectosteel enough. Plectosteel, you should highlight the only plectosteel and rest of the common. Rest of the means epidermis common character and cortex common character, but steel is plectosteel. That's it. In practical, but theory, you should write all. Actinosteel, you should write, and mixed protosteel, you should write, and plectosteel, and actinosteel, and protosteel, aplosteel. They are all you should write. Right. After internal structure, this a leaf anatomy, it is not important in theory as well as practical also. Don't ignore it. Reproduction. Actually, reproduction may be three types: vegetative reproduction, asexual reproduction, and sexual reproduction. Vegetative reproduction by body parts, sexual reproduction by spore formation, asexual reproduction by gamete fusion. Right? Here only we discuss about the spores, cone. You know how the cones are formed. Vegetative, leave it. Fragmentation means death and decay. That means if there is a lycopodium plant stem, it is break into pieces, and every piece can grow as a new lycopodium. That is called fragmentation. Hmm. Strobelus, okay, which should be clear. Strobelus or cone in lycopodium. What is strobelus or cone? Hello? Hello?
Reproductive organ which will
Right.
bear spores.
Right. Reproductive organ of the pteridophyta. Right. Let's discuss. Actually, we know two subgenera, Eurostachya and Rafalostachya. In Eurostachya, every leaf on the plant is a sporophyll. And Rafalostachya, the leaves near the apex bear the sporangia and are called sporophyll. That is the different. Eurostachya, there is most probably the entire plant with sporophyll. But in Rafalostachya, the normal leaves present at lower side, but upper side, apex species, sporophylls are present. Sometimes the sporophylls are distinctly smaller than the foliage leaves. For I already told you, foliage leaves larger and it is perform photosynthesis. Yesterday itself, I told you the aggregation of sporophylls together called as, hello? Aggregation of sporophyll, group of sporophyll. Hello? Hello?
Sporangium.
No. Group of sporophyll. Group of sporophyll. That means if you separate the cone, what are appeared?
A leaf and sporangium.
That is sporophyll. That means what I am asking. If you cone separated, first sporophyll appeared. When then you separate the sporophyll, you separated the sporangia and leaf. So the cone made up of by group of or aggregation of sporophylls. The cone has a central axis on which spirally arranged sporophylls are present here. This is common to every cone. So these are the cones. These are the cones at apical side. Position, maybe these are auxiliary position or dorsal of the sporophyll epiphyllus, subfoliar. Uh, these are the different position. If you take section cutting, longitudinal section, you can see in diagram. Uh, if you take a cone, this cone VS vertical section or longitudinal section appear like this. You must have seen in slide also. Actually, yesterday not completed. I told you to sir. Sir will explain to you. If you take the lycopodium cone, it is clearly have the axis like this. This is the axis center. This is axis center. Are you able to see line around?
Yes,
Hello?
sir.
Right. This is the axis. And the axis spiral arranged. Spiral means here the first leaf. This is 1.
Yeah, S shape, sir.
And this is 2, 3, 4. This is leaf. This is sporangia. Above side sporangia present. Right? So this is total called as sporophyll. This is sporophyll. Means leaf with sporangia. Leaf with sporangia. So that's why called as microsporophyll. Now arrangement also clear. Now let's discuss about. So this is longitudinal section already given practical, and also it is also important and theory also. You can yearly clearly given axis of strobilus here. Are you able to see marker? Hello? Hello? Hello?
Yes, sir.
Able to see marker? Cursor?
Yes, sir.
Right. This is axis, center axis, and the center spiral arranged microsporophyll. This is leaf with microsporangia. Leaf with microsporangia. If you see here, microsporophyll leaf with sporangia. At upper side, adaxial side, important. Where the sporangia present at leaf upper side, not lower side, upper or adaxial upper side, abaxial lower side, adaxial upper side. Now, sporophyll completed. Structure of sporangia. Sporangia, if you take the sporangia rainiform or kidney shape, it has stalk and capsule. This is stalk and it is capsule. Capsule is unilacular. Sporangial wall is 3 layered. Innermost layer is act as tapetum. Let's draw a simple diagram to you. For example, in clear view, this is the leaf upper side. sporangia presenter. This is sporangia. Uh, if you separate the sporangia, this sporangia actually attached to leaf with the stalk. So you clear sporangia stalk. This is sporangia. Actually, how many layers sporangia? Hello, number of layer. Sorry, 3 layer, two, 3 layers. So innermost layer act as a tapetum. What is the function of tapetum? Tapetum always give nutrition to the developing spores. So this is inner one tapetum. This one tapetum, and the innermost tapetum it insists sporogenous tissue. Mother cell undergoes to meiosis and produce tetrad and spores. Sporogenous homospores. That means actually first remember the function of tapetum. Tapetum actually before development of the spores, sporogenous tissue, just group of tissue. They are later developed as the spore mother cell. After the spore mother cell undergoes to meiotic division. After that, spores are formed. For developing all the stages, they require nutrition. So this nutrition provided by tapetum cell. So anyway, after formation, the all are similar spores. All spores what are produced, there are similar spores. That's why these are called homospores or homospores. Homospores. If it is different, that means one is small and some are large. That is called heterospores. So anyway, it is homosporous condition. Next, what happened after formation spores? Hello. After development of spores,
They form gametophyte.
right. Let's see structure of spores. Already discussed homospores. This is all are similar. They are actually formed as tetrad. Tetrad means four as a group. Tetrahedral shape. This is range that is not important, and each and every spore triradiate ridge and two-layer exine and intine. This is triradiate. You can see in diagram triradiate ridges. Triradiate ridges and made up of two-layer, smooth or variously sculptured outline, haploid nucleus. That means these spores are haploid. Cytoplasm filled with reserved food material. These are utilized for germination. How it is liberated out? Dehiscency. Elongation of the internodes, strobilus, sporophylls are spread out, exposing sporangia. When the cone or maturity stage, automatically the dehiscency occur as a result from the sporangia, spores are released out. Now after take the sufficient place, before that they are disseminated sufficient area. Later they are take sufficient plate and grow as a germinate as a gametophytic plant. Germination of spore. No need to write all this. After germination, gametophyte formed, right? Isn't it? Hello?
Yes, sir.
Right. There are 3 types of gametophyte. Another name prothali. Don't get confusing this word. Gametophyte or prothali. These are 3 types. Cernum type and clavatum type, phlegmaria type. That is the based on the species. But remember, these are all belongs to gametophyte or prothali. Let's see one by one. That is the based on shape and structure. Cernatum type, cernum, sir, craniatum type, cranum type. It is electric, erect, and cylindrical. Let's see, like this example. erect and cylindrical, grow surface ground, colorless, basal portion buried in soil, uh, generative zone green, six organs at the base of the lobes. You know already what are the six organs here. Anthridia, archegonia. Rhizoids are restricted to lower buried side. Endophytic fungus is present in the basal portion. That is important. Inside the prothali, the fungus also get associated. Meristematic tissue present as rim and prothallus independent preparation of food material. Their own. That is not a dependent. Already discussed yesterday. So based on shape, already told you this is erect and cylindrical. One is. Next one is this one. Sternum. Longitudinal section. Clavitan type. If you see the clavitan type with the diagram, this one. This is clavitum love shape, heart shape, pygmalion type, and what are they given? So any type. Prothallus, now long here the prothalli fleshy, not fleshy or non-green, total saprophytic and completely subterranean and perennial in nature. Actually, the gametophyte, as we know, they are the autotrophic, but clavatin type is different. It is not a autotrophic, it is fleshy and non-green. Non-green means no chlorophyll, totally saprophytic. What is saprophytic? Hello?
Living on dead and decay.
Right, death and decay. So clavatin type is not a autotrophic, it is saprophytic type. Uh, but anyway, at the final, when reach such maturity stage, it consisting the six organs, both anthridia, archegonia. This is second type. We can see here the anthridia also, where the anthridia, here the anthridia, and these are the archegonia after fertilization, embryo formation here. This is the structure. And phlegmaria type. A phlegmaria type, this is the prothali or aerial, but saprophytic in nature. Grow in tree trunk. This is also saprophytic. Below the coating of humus, this type of found in epiphytic species, Lycopodium, example Lycopodium phlegmaria. The spare germination immediate and gametophyte grows only one season, but not all season, only in particular season. Prothallus consists of tuberous central part, which number of colorless slender cylindrical branches, irregular fashion. So that is like this. So irregular branches are present, like variety. And this is also saprophytic, last like a second one. Branches bear the sex organs. These branches consisting sex organs. You can see all. So anyway, the prothali again given prothali consisting anthridium, archegonium, anthridium structure and archegonium structure and fertilization. After what is fertilization? Already you know this. Hello,
Fusion of male and female
right,
gametes.
right. After that, what happened?
Zygote is formed.
Zygote is formed. Zygote develop embryo. Embryo further developing this lycopodium sporophyte. That's it. Draw this
Yes.
life cycle of lycopodium. Draw that means you just write names. Like first instead of this diagram, lycopodium sporophyte. Next from the lycopodium sporophyte parts root, shoot, and leaf. Next cone, sporophyll, sporangia, spores like this. These spores. Next this gametophyte or prothali. From prothali embryo. Embryo again developed as a sporophyte like this. Draw this. Take picture. Okay.
Yes, sir. Yes.
Right. And any doubt in lycopodium? Section cutting completed. Was it explained or not? Hello? Actually, today I feel feverish and some type of cold and cough. That's why I'm unable to attend practical classes, unable to take. And my duty responsibility to hand over my colleague, Dr. Ramakrishna sir. He will take. Was it explained or not? Hello?
Sir, we did equisidum section cutting. Lycopodium we did not do.
Okay, no problem. Actually, the material not available, I think. Whatever it may be from Pteridophyta, one
Yeah,
enough.
yeah, yeah.
But
Equisetum, yeah.
right. But point of theory, we explained all.
Yeah.
Explained Lycopodium we will explain, and Equisetum explained, Marsil explained, Pinus explained. We will explain Needham also. But that is theory purpose. But in practical purpose, most probably from Pteridophyta one, from Gymnosperms one. But in the exam, in practical examination, we will give any one. Okay,
Yes.
right. And now let's take another one. Uh, Equisetum. Now tell me, Equisetum sporophyte structure already discussed yesterday. What you learned already you discussed yesterday, and you have seen the specimen also, right?
Yes, sir.
Hmm.
Equisetum is also called horsetail.
Right,
And this also same like similar to uh, Lycopodium.
right. So that means some similar character, but not exactly. Some, are you
Yes.
able to see the Equisetum slide?
Yes.
Right. This is just take a picture. This distribution habit not important. Class Calamiphyta, Equisetails, Equisetum, genus Selaginella, constituent 25 pieces, composition, distribution. Equisetum grow variety of habitats. The distribution that is not explainable. This is only you can read, you can understand yourself. Equisetum monoecious. What is monoecious?
Both
What?
the male and female are present in single plant.
Right, right. No, sporophytes. This is important here. Morphology already discussed yesterday. If you see the sporophytic plant body, it's not important one. Here the sporophytic plant body differentiate root and shoot and leaf and rhizome, right? The root they develop from the node of the rhizome base of stem. They are long, slender, adventitious. Function
Yes.
already know. What about the stem? Stem from rhizome, aerial shoot arise toward the upper side. They generally branched. Aerial shoot two types, sterile and fertile. What is sterile? What is fertile? Hello?
Yeah, sir. In sterile, it will not have reproductive properties.
That's it. That's it. Fertile have the core. That's simple. And leaves, here the leaves are very minute and scaly leaves, thin and nerved. This present from the whole, or we can see. Already we have seen yesterday specimen also. The brown color around the node, brown color structure you must have seen yesterday, right?
Yes. Yes.
Right. So these are the scale reduced leaves. They are present, but there is no clear foliage leaves. Lower and each leaf unit forms sheath and node, rhizome, stem, horizontal stem called as rhizome. It is creeping, well branched with nodes and internodes. On its nodes present scaly leaves. The scale is small, slender, more or less unity. It forms the sheath around the node already discussed yesterday. Internode functions of the different parts, root function, rhizome, sterile shoot is not an important one. Already, this the structure also you
Yes.
must have seen yesterday. No,
Yes, sir.
don't discuss. And remember, as you said, it is another name horse tail because of the
Yes.
tail, the plant similar to the tail of the horse. So already this all completed. Stem completed, leaves completed, root, rhizophore. These are all completed. If you want, take picture. This
Yeah.
sporophyte.
Sir, this PDF will not be uploaded on the website.
I will send. Do you have group?
I don't know, sir, which group you are talking. I am having only students group, not.
Yes, a simple type like ecosystem PDF in Google and search it. They can. So many PDFs are available.
Okay, sir.
So this is structure, anatomy of stem, right? Already told you in anatomy, internal structure, three parts. What are the three parts?
Epidermis, cortex, stem.
Already, I think it was explained today.
Yes, sir.
Should I explain again or so? Anyway, let's
No,
complete
sir.
it. Two minutes enough. So
Okay.
already you know this three epidermis and cortex and endodermis and pericycle and steel. These three region. Sorry.
Identification characters are furrow and ridgy epidermis. It
Right.
has
That is the in epidermis. Actually, epidermis outermost layer, single layer, but it is furrows and ridges are present in epidermis. That is one of the character. Here the cortex,
stomata are present in cortex
right,
and basilar
uh,
canals.
from highly differentiated, that not a homogeneous, heterogeneous. Already told you, homogeneous means one type of tissue, but here different type of tissue. That mean inside the ridges, each patch of sclerenchyma present and followed by chlorenchyma tissue. Sclerenchyma give mechanical strength to the stem. Chlorenchyma, already you know that it is perform photosynthesis.
Yes.
Okay, and just inside the each groove, that is important character. Just inside the each groove, the large air canal in the parenchymatous cortex. This is called valvular canal. This is very important. Inside the groove, large valvular canal present. Uh, Endodermis, outermost layer without cellular space, single layer, pericycle, not important one. Inside the steel, vascular bundle present. Inside, actually, steel, later, uh, there is pericycle is given. Actually, before pericycle, the put heading steel. In steel,
Yeah.
pericycle, it is the first layer or inside the endodermis. It is first layer of the steel. Pericycle is first layer of the steel or inside the endodermis, present single layer pericycle. This is not clearly present in the all the species. So pericycle not clear. So it is not important in this ecosystem. Next vascular bundle. Here the important thing is the vascular bundle type is siphonostelic type. What about in lycopodium? Lycopodium protostele.
Yes.
Here siphonostele. Siphonostele means is a ring with pith. Pith present, medulla present, that is siphonostele. Already I have drawn just
Yes.
before. That is siphonostelic. With pith, medulla, siphonostelic. Without pith, protostele. Simple.
Okay, sir.
So they are in a ring present below ridges, alternate with the valvular canal. This important valvular canal alternate. Uh, so center pith is present like this. This is ridges, furrows,
Yeah, sir. We
sclerenchyma,
saw this just
alternate
now in the
chlorenchyma.
microscope.
Right. This is valvular canal. You can see here valvular canal also. In ground plan, you can see directly clearly. This is valvular canal. The alternate vascular bundle. In diagram, in theory, practical also, you should draw like this.
Okay, sir.
This is valvular canal, alternate, not opposite. Alternate
Okay.
vascular bundle. Valvular
Okay,
canal,
sir.
alternate. That means between vascular
Yeah,
bundle,
yeah.
between valvular canal, vascular
And
bundle present.
yes.
That's it. And inside the vascular bundle, carinal canal. This is also another important. Inside the vascular bundle, inside exactly xylem, the carinal canal also present. Carinal canal. This is, you can see here, you can see the dot-like structure in xylem. This is
Yes.
called carinal canal. And central pith, central cavity or medulla. Anatomy of rhizome, no need. Anatomy root also no need. No need means it is not important in theory as well as practical also. Reproduction. You'll mainly focus on sexual reproduction by the formation of spores. How
Yes,
the
sir.
spores are produced? Hello?
Yes, sir.
Spores are produced inside
Spores are inside cone.
cone. That's it. Cone is the compact structure, terminal axis. This inside the cone, sporangia are also present. Structure of cone. Each cone central axis. This axis umbrella says sporangia 4. Here important thing one is actually uh, so what are they given here? Sporangia 4. Actually, instead of sporangia 4, actually in Lycopodium, what is present? Sporophyll. Sporophyll.
Yes.
When you discussing about the sporophyll structure, did you discuss about leaf in eczytem? Did you find out leaf, clear leaf in eczytem?
No, sir.
No, that's why there is no sporophyll. Instead of that, only sporangia with 4. Sporangia with 4. That is called sporangia 4. Each sporangia 4 is stalked and sporangia bearing organ, the free end which flattened and peltate like disc, hexagonal structure, protecting covering, annulus. They are the part of this. And so let's discuss one by. Let's see the first diagrammatically. I will explain with the diagram, but just listen here. The disc hexagonal shape, protective covering, the flattened tip, protecting covering present around the sporangia, and below the each tubulus present the calyx-like structure called annulus, which is also protection function. So here, so this is the strobylus or cone. This is annulus. Cone. This is annulus as a ring. If you separate the cone, if we take a pelted disc, this is disc. Disc either inside two to sporangia raised. Inside the sporangia, spores are present. Each disc with stalk. This is one sporangiopore. Like this, if you take the entire longitudinal section, the diagram would be this is only one sporangiopore. Now let's take right. This is stalk axis. Axis and the axis. This is stalk. From the axis, stalk erased. Stalk filled disc like this. And either side, how many sporangia are there? How many sporangia are hanging here? Either side. Hello? Hello?
Yes, sir.
Are you there? How many sporangia hanging in the pelted disc here? Either side. This is the axis, red color. First, this is axis.
90.
Uh,
No, no.
no, this is the one pelted disc. In one disc, how many sporangia?
2.
Right, two sporangia present. That's it. No leaf. The only sporangia 4. Instead of sporophyll, sporangia 4 present. That's it. But in Lycopodium, the same place with the leaf sporangia. But in
Yes, that's why
ecdysium with a yes,
sporophyll.
sporophyll. Here no leaf, only 4 with sporangia. That's why sporangia 4. That's it.
Yes, sir.
You know
Sir,
the rest.
it's lunchtime. Can I leave?
Okay. Just 1 minute. 1 minute. Can I continue? Should I continue?
Yeah, you just ended. Then I leave.
Okay. So the rest of the matter you know already. So what happened after descent of the sporangia? Spores are released out, right? Hello?
Yes, sir.
Spores are released out. These spores further germinate into
Uh, Gametophyte.
right gametophyte. The gametophyte again produce what happened here? Is gametophyte again produce the sex organs such as
Yeah, Archegonia and Anther.
right. What happened here? Right. This is also homosporous. Remember, homosporous, heterosporous means homosporous means similar spores. Heterosporous means different. And here the another important character. Here the spores are covered by small thin elaters. Elaters, what already you listen in Markantia. So in this also spore with the elaters present. These are helpful for spore dispersal. Inside the sporangia, elaters present and gametophytic space you know already. This is gametophyte. Already here the antheridia, archegonia, fertilization.
Yeah, sir. Similar.
Right, similar, no changes. Similar, no changes. Right. Take this life cycle. Take picture this life cycle.
Yeah.
No
Sir.
need diagram, just diagram it with the matter
Yeah.
enough.
Yes.
Take this picture. And we will
Thank you, sir.
discuss tomorrow about Marsil. Right?
Okay, sir. Yeah.
And please attend. Please attend.
Okay, sir.
Thank you all.
Thank you, sir.